In this lecture, I will talk about local alignment. First, let's review the last slide from last lecture. So here we got a global alignment result and a user-specific scoring matrix using Needleman-Wurtz algorithm. And when we track it back, we start from the lower right corner and ends at the 0, 0 point in the upper left corner. In other words, we align all the bases of the two sequences from the beginning to the end. This is so-called global alignment. As mentioned earlier, Needleman-Wurtz algorithm can find the optimal alignment spans the complete sequences end to end. This is appropriate for closely related sequences that are similar in length, and it has been widely used in early comparison analysis of proteins. However, as more and more sequence data generated, the Needleman-Wurtz algorithm for global alignment has encountered some problems. First, as more and more protein sequences were determined, researchers found that although functional related proteins may differ widely in overall sequence, but often they have the same function domain. Protein domains can independently play a specific biological function and are quite conserved among different proteins, but it's obviously impossible to find such a subregion only by global alignment. On the other hand, in 1970s, intron was discovered. People started to know that the gene has introns, and the intron's mutation rate is very high. In fact, it doesn't matter. The intron doesn't matter. What matters is the quality of the alignment of the exon. So how do you deal with such a problem that a sequence with important intervals and not that important intervals? What do you do? The development in biology, the discovery of protein domain and the DNA intron raised some computational questions, and people began to realize in early 1980s that we need new ways to find similar local sequences. In other words, we need to do local alignment. So in 1981, the physicist Temple Smith and the mathematician Michael Waterman published a 4-page article in the Journal of Molecular Biology. In their paper, the Needleman-Wurtz algorithm is improved, and a local alignment algorithm called Smith-Waterman is proposed. This paper has been cited more than 8,000 times so far. It's one of the most cited papers in the field of bioinformatics. And in this paper, Smith and Waterman proposed that a local alignment of two sequences allow arbitrary length segments of each sequence to be aligned, and with no penalty for the unaligned portion of the sequence. Otherwise, the score for a local alignment is calculated the same way as that for a global alignment. So they just did a very smart trick based on the global alignment, and it turns out that this is very useful. Okay, let's look at the algorithm first. This is the algorithm for the global alignment, and the Smith-Waterman local alignment algorithm. Just add a 0 to it. That is, at each iteration, a lower limit is added to the alignment score. And this 0 has 3 impacts. First, it allows a node to start at 0. This is very important. And the second, the score for a particular cell cannot be negative. If all the other score options, 1, 2, 3, produce a negative value, then a 0 must be inserted in the cell. Third, record the highest score node and trace it back from there. It not has to the tracing back not has to be the lower right node. It can be any node in the matrix, that as long as the node has the highest score. So the alignment is not has to be started from the beginning, not has to be ended in the end of the sequence. We still use the example in the global alignment section to demonstrate local alignment algorithm. Here is exactly the same as before. Same score matrix, same sequences to be compared, except that 0 is added into the iterative function of the dynamic programming. So we still fill in from both sides. And in the global alignment, all those numbers are negative. And now in local alignment, we do not allow negative numbers. They are all zeros instead. Then we start filling the middle box. Then we look for the highest score nodes. We found there are 2 highest score nodes, 2 here and 2 here. Trace back, and we got the first alignment, DP, and trace the second highest score nodes back. We got another alignment. We can see that the first alignment is a local alignment and the second alignment is a global alignment. Here I want to show you another example, 2 DNA sequences. The local alignment result is shown here. We can find more differences comparing to global alignments. First of all, unlike the global alignment, local alignment now have many independent passes here. At least I have shown 3 different passes. The blue one is the optimal pass. It has the highest score 4. And the 2 red one is the suboptimal alignment. It has the not the highest score. It has a score 3 or 2. This is because according to the modified iteration formula, if 3 possible alignments get a score less than 0, then 0 is taken. Then in other words, the previous iteration path was terminated at 0. Therefore, the starting point of the traceback not necessary to be the lower right corner, and not necessary to be, and the ending point not necessary to be the upper left corner. So in fact, at this time we can see that all the backtracking is local, and the resulting alignment is also local. Therefore, unlike a global comparison, we can also get a suboptimal local alignment in addition to the optimal alignment. Okay, to summarize, the Smith-Waterman algorithm does a simple adjustment to the global alignment iterative formula by introducing a lower limit 0 for the alignment score. This lower limit essentially provides the ability to restart the alignment after regions with large differences. Therefore, the similarity at the local level can be effectively evaluated.